our loving ones. Right. Before going to the depth of the title, I want to brief about the Dabur role. Yeah. As a Dabur India Limited company is a very well-known company, FMCG company in the globally. And in this segment, from the R&D point of view, we are dealing in the three segments. First segment is healthcare products, in which we deal about our Ayurvedic products, classical products, and our proprietary medicines in this, which is totally based on the herb medicinal plants. And the second one is the personal care and the home care products. In this, we are the good quality products like the Fem is our sister concern is there, and O'Donnell. There are a number of the brands are there in this particular category. Dabur Amla is a very well-known category in this particular personal care and the home care products. And next one is the food, which is I'm leading right now. The food beverages, real juice, honey segments. There are a number of the variety products we have. We explored the lots of segments in this particular category right now. And as a R&D, we have the depth role to investigate the NPDs, US stability studies. There are lots of things comes under the R&D job profile point of view, I'm saying. In the same Dabur Research and Development Center, there is a main term you can say this is the regulatory department. As we are dealing in the globally, then we have to meet all the regulations parts in the worldwide. That's a separate regulatory team with us, which inspect the regulatory guidelines of all the countries and suggest what's the limits we have to follow for to sell in the particular country. And the next one is the Bioresource and Development Center. This is the main department in our DRDC center, which is and their role is meant to purchase your herbs, and they also in the contract farming, and they also involved in the number of the projects to avail the best medicine plants to our Dabur premises to do the best quality products. In short, I briefing somewhat about our role in this. What happen for any department, for example, either it is a healthcare department, either it is your personal care department, either it is a food department. They do your NPD. We standardize their specification parameters, right? Which includes some critical parameters, some include some non-critical parameters. Critical parameters include like microbial load, high metals, right? Like your active components in your NPD, right? And with the time, we have to study out that during this whole shelf life. If the product is found stable during the whole shelf life, then we go to the for the production point of view, right? We set the standardization parameter. They will check at each and every batches in the production plant. After that, only it goes to the market. That's simple. Beside of that one, we also do some benchmark study, investigative analysis. Your vendor development part is our major role is there. Vendor development part includes the standardization of the raw material. So to provide a good quality check on the each and every raw material which procured from the Dabur end. We always neglect the superior quality of the Ayurvedic herbs in from the surroundings, right? That's the main. The backbone of any industry is always its R&D center. This is the overall view of how we work, how we handle all the segments there. Mutually, we do work with all the departments, your pharmaceutical team, your regulatory team, and the other departments. And then we take the mutually call on that one, right? So now comes on my topic today is the quality of herbs. Quality of the herbs is simple what you can say. This determine the purity of your drugs, right? Its contents of the active moieties in your herbs is there, and to find out their chemical and physical stability of that particular herbs, right? And how it is possible, it's the biggest task is there. But by help of this chromatographic technique, why I choose that one category, because it is you can be very robust, sensitive, and accurate to check out their purity at the ground level. So in the first figure, I shown the SPLC chromatogram, which shows the number your carbohydrate moieties in sugar product base, and the last one is this is the simple SPTLC profile to screen out the phytoconstituents. I will brief each and every this chromatography technique in my next slides, but in short, because each and every category is comprises the whole things in that. That's why I will summarize the little by short. Now, I am described in the chromatography. It is simply a separation technique. It's include a separation bed, which is a silica bed, versus there, and the column chemistry you can say, right? And your analyte, which is we extract out from your house. Then I will slow down this space. Then I'll brief you something about that one, how it's work actually. In the chromatography, first we extract out the active moieties from the husk by using the any suitable solvents, right? After the extraction of the solvent, we inject that particular your sample in the your any of the chromatography techniques. That include your this is SPLC, high performance liquid chromatography. That is equipped with number of the detectors based on the nature of the your moiety. For example, if it is a chromophoric compound, then there is a need of the PDA photodiode diode array detector, and the simple UV detector you can use, which is commonly used for the work on the two wavelengths. And if it is your compound is a non-chromophoric, right? Like your carbohydrate moieties, and like your glucose, fructose, sucrose is basically present in the honey and the other matrix also. We can check out with the help of the refractive index detector. So there's a very specialties in the detection point of view, I'm saying. So we have to select out the particular detector based on the our analyte, simple. And in the SPLC, there's a one of the high sensitive technique. After that one is the LCMS. LCMS is the liquid chromatography equipped with mass spectrometer. You can just single quad pole. For example, in the chromatography, what you seen, I will just briefing about the SPLC. Right, it is simply based on the separation technique. We identify the analyte based on the retention time of your reference standard. Right, yeah, but in the natural point of view, I'm saying in the herbs, there are a number of the phytoconstituents are there. There are not a single molecule is there which is responsible for the phytoactivity of the compound. Yeah, the number of the phytoconstituents when we inject in the SPLC, they get separated out, right? But some molecules may behave similarly. May they have the same polarity, may they have the same partition coefficient. That's creates some your doubt in your mind from the purity point of view. Then there's a need of the higher sensitive technique. This is the LCMS. It based on the how is it work simply. In this, we inject the urine light after passing through the column in the mass spectrometer. Right, it goes to the mass spectrometer that ignite with the help of the source energy, ionized in the and then in the collision cell it can collide and give the fragments ions, right? On that virtue of that one, we get some parent ion which we can identify from the your analyte. A simple identification technique is there, but itself is a very broad one. So I'm just briefing this short how is it work, how it is important, and how is beneficial to sustain the quality of the your house, right? And this is the same simply LCMS MS liquid chromatography equipped with the triple quadrupole. This is the highly sensitive, and this is mainly used for the identification, quantification of the marker compounds of the your house. For example, ashwagandha, there is a bidon lights, we can quantify with the help of this one. In the Indian pharmacopoeia, there's a two methods are mentioned. One is the gravimetric method, but SPLC is the more sensitive one because you in the gravimetric method, there's a more number of the misleading compounds may occur, comes out that, but it is a very precise technique SPLC. So we definitely go ahead with the HPLC profiling only, right? And this is the next one is the gas chromatography. This is simply mainly used for the volatile compounds, right? In our R&D, this is a very useful technique. It is mainly used for the pesticide analysis. Also, there are the volatile compounds like the chlor, eugenol, thymol as well. And there are a number of the volatile compounds are there, which we need to analyze time to time to check out the stability of the products, right? And similarly, like the SPLC, there the number of detectors are also in the gas chromatography also. This is the RFID flame ionization detector. Instead of the flame ionization detector, for the more sensitive point of view, you can use the MS mass spectrometer, single and the triple quadrupole. This is totally based on the analyte chemistry, simple. And this is concentration in your matrix. And the next one is the high performance thin layer chromatography, SPTLC. It is main you can say the very good technique which is mainly used to standardize your phytoconstituents present in your house, right? How's it work? Like the finger. Why we are saying the fingerprinting profile? Like the fingerprint of the any person cannot be matched with the other one. Seen the similar way, the fingerprinting profile which is developed for the herbs in a particular solvent, in the particular extraction media, and all these things, it cannot match with the other ones. However, there are some exceptions are there, no doubt. In this, we can relate on the based on some higher techniques. But it's very useful technique from the herbal product point of view, I'm saying, right? For example, just I'm giving one example of that one, how it work actually. In the NPDs, new product development point of view, I'm saying, right, when any entities comes to us to standardize their parameters, we put the SPTLC there to screen out the phytoconstituent profile of the product, right? We measure at the initial level, then further at the accelerated studies, when the samples comes out of the third month in the accelerated conditions, we check out in a constant way without initial profile. If you found any deviation in the some spots are missing like this, that gives us some hint to you how's your product behave, overall behavior you can say. Right. That's why it's a very, very, very important technique in the chromatography point of view to especially to standardize the best quality herbs in the from the market, because there's a number of spurious drugs out there. You cannot rely based on the only single compounds. You can say the single marker compounds. You can say, right. So overall profiling is very must to standardize the you can say the sustain the quality of the herbs. Right. In the fourth one, this is the part of the SPTLC. This is the one device TLCMS interface, which I will explain in my next slides. Actually, it is very wonderful technique, very commonly used to standardize and to isolate the compounds from the TLC. And I will explain the next slides. It is very important from our R&D point of view. There is a number of the products, and I will explain the next slides. And fifth one, I explained the some higher techniques, very sensitive as compared to all the HRMS, high resolution mass spectrometer. Select the SPLC, LC chromatography equipped with the high resolution mass spectrometer. Here, the precision level of the parent ion, M by Z ratio, you can say, is very high. Up to the, you can see the 4 decimal point, you are getting that accuracy level there. So, from the accuracy point of view, from precision point of view, we are using that technique, but it's very, very costly. So, we cannot implement for all the each and every techniques. That's why. So, we have to rely on the first. So, there's in the R&D, there's some steps are there. Go with the most commonly technique. If we found some difficulties, then go to the next level, like the most sensitive technique, and then the next steps to us. This is totally based on your expertise, your judgment power. You can say you have to select out your technique. Simple version I'm saying. Right. This is the wholesome chromatography technique, which is most commonly used in R&D centers. Other than this, there's a number of techniques also. Chromatography techniques are there. Size exclusion chromatography, column chromatography. There are a number of techniques are there, right? But I'm just more explaining here the most common techniques, right? Then how's this techniques helping you helping the to sustain the best quality? How does it work? That's why I'm quoting some examples in the next slides. That's simple. And before going to the in the tab, there's a need to understand the basic approach of the chromatography techniques. Before, if you have some targets, then and then you can achieve more in your life. Simply in the same things, if you have to set your targets, this is more achievable from the quality point of view. That's why I'm saying in the chromatography technique there are two approaches are there. One is the target approach. Target approach means to quantify the target moiety. For example, in a Sagunda, when the light, Google, there are glycerols are there. These are target moiety. You consider your marker compounds. But in the herbs, there the number of the other compounds are also there, which we can evaluate by the virtue of non-target approach, right? Non-target approach, that means we are not targeting the any particular molecules there. Beside of your marker compounds, we are targeting other compounds present in your herbs, because it is a you can the synchronization effect is there. Not the single marker compound is responsible for the activity of any plant, but they need the other compounds also. That's why I'm saying nowadays the concept of target and non-target approach is going very, very, very well to sustain the best quality of the herbs for the future point of view. I'm saying that's why it is a simple, very good technique to check out to identify or to quantify the secondary metabolites present in your herbs. That's you to check out the two approach there, target and the non-target in your mind. Simple. And on the basis of this target approaches, I am just explaining the next words, which is very commonly used nowadays to evaluate the quality of the herbs, which is that metabolic profiling. Metabolic profiling, that's simple what I'm saying. This is a simply identification or the quantification of all the maximum secondary metabolites present in your herbs. Right. This is very novel concept is there. Number of the papers are there. Just I'm showing them my one of the paper which I done on my PhD research. That's simply the evaluation of the geographical reasons for the best quality of the Google. You know the Google is a very endangered species is there right now. So how to evaluate that one? You can evaluate by the virtue of your metabolic profiling. Right. That's why this is the metabolic profiling of the Commiphora viti, Bhandari bark, oligum resin, and the stem collected from the different region of India. I just briefing that my research paper there. How's your chromatography technique help to you evaluate the best quality herbs and their part also, which is marked as a therapeutic active part point I was saying. Right. In this study, what I did that I explored the other parts of the Google instead of the oleogum resin. Oleogum resin is the main active part of the your oleogum resin. This is the extract out from the your Google plant after the incision on the Google. And the number of the papers are there on the Google. It's a very well known well-known medicinal quality herbs is there, right? But there's a need of how is to let some other parts also, right? And that's why we compare the stem part. What I did then, I pulled out their stem part from that the main bit of the Google, then screened out their secondary metabolites with comparison to the our control, which is your oligomycin resin. Then we study out that oligomycin resin with respect to the stem, and we have very interesting finding we found in this actually. And you can imagine that in the bark part also there is a number of the phytoconstituents which match similarly with the your oligomycin resin. So we can also explore your extract to the your bark for the if it is a shortage of the oil in this oleum resin in the market. So we are giving the more option to the market. That's why this also is only done with the help of the chromatograph techniques. I use the SVTLC there. I use the LCMS there. I use the SPLCs there to standardize the this quality of the herbs in the main compounds. So that this particular Google, very beautiful paper is there to how we summarize the things with the help of chromatography techniques there. Now, now there's a number of the endangered species in the market, right? You can say that you can procure as a crora, you can say Google is also an endangered species. Then how to procure that one for the to meet the future demands? This is the need of how is there to maintain the best quality medicinal plant for the future point of view, right? So on that prospectus, we have a separate department is there, Bioresource Development Center is there, which basically involved in the procurement of the your best quality herbs and involved in the number of the contract farming, involved in the number of the government projects. And we have also a tissue culture center in our lab, where we develop some tissue culture. So the best quality from the origin, best quality origin herbs. And we check out there and the concentration of the market active compounds with the time in that. Here I just showing some the culture of the Google is there, where we develop some Google Callus in-house. And then after that, we standardize that one with the help of the different techniques. You can say by the LCMS or PLC, you can check out that with the one. Right, in the simple, in the downstairs sides, I'm just showing the metabolic profiling of the LCMS. And this next one is the marker-based study of your callus, mainly to detect the gallstones present in your callus mount. So it is very good techniques. We're involved in the each and every steps to sustain the best quality of for the future point of view. And this is the very, you can say the DNA sequencing, how does it work, right? In our R&D center, we are also doing the DNA sequencing of our number of the species to protect our products from the superior low quality herbs, right? So we adding that parameter in our each and every specification parameters in the for the especially for the drugs which are too costly is there, right? Right. So because of the due to the high amount of the your these drugs, there's a lots of chances of the substitutions there to how to protect that one. We enter a end checkpoint there by the DNA sequencing. We do the DNA sequencing of the particular species and match with the each and every entry for the critical plants. You can say only right. So it is a very important techniques basically. And the next one, I'm showing the one of the research paper. That is how to geographical lesson of the best quality house from the geographical. For example, one house is which is propagated in all over the India, out of the other countries also. Then how to check out the quality of the house. So by virtue of these techniques, chromatographic techniques, we can check the concentration of the active moieties in that one. We can do some metabolic profiling also. We can do you can the TLC fingerprinting profiling also, right? So that's why I'm showing one of the paper to just give an idea to all of you that how it work actually, right? That's the simply I done one of the study on the Google. That's my research paper is there. Effect of aging. That means the how's the for example, I'm just speaking about the Google oligom resin, which is executed from the Google. It is comes with the, after the maturity of the 5 years, right? Not before that one. Just after the maturity of the 5 years. My object was there to, at which age group to select out the inside the plant, because after the incision there is less chances of survival of the plants. That we do very precise on that one. Then we related that one to impact of the impact of phasing on the concentration of glustrons. And then we check out the different geographical regions Google found in the India, and we check out the concentration of that one. From my point of view, the Kutch origin Google is a very good qualities, give the good quality of the oligom resins, which have the high concentration of the glustrons and giants, which we screened by the metabolomic profiling also. That's why it is very nice to check out the concentration of each and every geographical origins to get the best quality products, right? So in the next slides, I'm just giving the one of the hint of the TLCMS interface. How does it work? Actually, how it is increasing the demand side now in the industries. That's a simple words, and but it is a part of the SPTLC, high performance thin layer chromatography. Uh, After the development of the TLC, when you apply your extracted solvent on the sample on the TLC, then after the development part, then you get the very good result fingerprinting profile of that one. And if you want to extract out the particular spot to check out the unknown moiety, right, to identify that particular moiety, you can use this TLCMS interface. Interface means that it's serve as a mediator between your TLC and the mass spectrometer. Simple, right? After the development of you can say I'm going the first step by step. After the application of your sample with the help of the sample applicator on the silica plate, then you develop your TLC plates there, right? And then you get a well-resolved fingerprinting profile of that one, and then you can put the plate on your TLCMS interface. Then you can, by the help of the interface, you have to focus on the particular your spots with the help of the laser beam is there on this. I will show in the next phase to the how it work actually. And then you, after the extraction of that particular moiety from the TLC plate to inject in the MS, then you get the mass spec of that one, and you correlate the things as per your assessments, right? And how it work actually, that's the very interesting thing is there. In the lower side, in the workflow, first one, the close view of the interface. What happened when you put the plate on the interface? You just select out the particular spot and put on the center of your beam, right? Then you put down the laser in the extraction head. When you put down the extraction head, the mobile phase, which is comes out from your SPLC pump, comes down and extract out the particular moiety on the TLC plate and filtered out from the outlet, and which pass to your mass spectrometer. From that, you can your purity of the compound. What compound is there, molecular mass, what's out there. That's give us some idea about that one. Very interesting techniques right now. We are using that one. The wholesome overall, the chromatographic give best deal in the selection of the best quality house. That's by proper use of that chromatograph technique, you can sustain the best quality house. You can do the best quality of the products for the market. That's where to more concise on your practicality to how to ensure the best quality house for the fish point of view. It's a very helping tool for the industry point of view, I'm saying. You can explore the academic also. There's a number of academics institutes are there which are using that one techniques, and the number of the research papers on that one. And this is in the short from my side. Just thanks all of you. And this is my contact number for the